My name is Olga Lazareva, Technical University of Munich. My talk title is BiCoN, Network Constrained by Clustering of Patients and Omics Data. Let's first discuss why do we need network constrained by clustering. When it comes to biomarker discovery, supervised methods are often used. And even though biomarker discovery has greatly benefited from it, supervised methods can be strongly biased by our current understanding of diseases. And that's the reason why unsupervised methods are getting more and more popular in the community. On this slide, we see the most common applications of clustering methods, which are discovery of data-driven patient groups, or gene clustering, or biclustering, which is often used to discover both patient and gene clusters at the same time. And when we want to find gene groups based on their relationships in a network, community discovery methods are usually applied. But we like approaches that can integrate our knowledge about functional relationships of genes in networks and expression profiles of patients. To bridge this gap, we develop BiCoN that takes as an input a gene expression matrix and an interaction network and selects genes which are the most valuable for patient clustering, while also connected in the PPI network. A simple schema of the framework looks like this. BiCoN constructs a bipartite graph from gene expression matrix and then adds edges between genes based on a PPI network. Then we use a combination of ant colony optimization and local search algorithms for robust and fast subnetwork extraction and patient clustering. Let's see now how this approach leads to computational advantages that BiCoN has. Here we wanted to introduce varying level of noise to a lung cancer dataset. We randomly selected between 0 and 90% of the genes and permuted the expression values. The left figure shows that the algorithm is able to recapture the cancer subtypes almost perfectly up to a noise level of 50%. The right figure shows a strong positive correlation between the objective function scores and Matthews correlation coefficient, which confirms that the objective function is high when cancer subtypes are well separated. The network interpolation forces BiCoN to select functionally connected genes even when the signal is disturbed by noise. Another important feature of BiCoN is its robustness to batch effects. Like in the previous scenario, we can see once again how the network integration allows BiCoN to be much more sensitive to the foreground gene signal, mimicking activity of a biological pathway. To demonstrate this, we simulated two datasets, such that the signal for a batch is twice as strong as the true biological signal. The genes that carry that true signal, we can also call them the foreground genes, were connected with one another in a simulated Barabasi-Albert graph. Then we normalized both batches, and now the batch effect is almost gone, but also the class differences are almost lost. Then we applied BiCoN, and as you can see, BiCoN has restored the true classes almost perfectly. Due to the fact that BiCoN also relies on networks, and networks do not have any support for batch effects in gene expression data, BiCoN is less sensitive to batch effects comparing to other methods. To prove this point, we have simulated 10 datasets with batch effect as previously described and evaluated the performance of 3 popular biclustering methods and 2 clustering methods. In the end, only one biclustering method has produced any results. Also, to make sure that we do not favor BiCoN by enforcing connectivity of the foreground genes, we also performed simulations with a Barabasi-Albert graph where foreground genes were randomly distributed and not necessarily connected. In both scenarios, BiCoN performs better than other methods, as you can see on both plots. Let's move on now to another important advantage of BiCoN, its contribution to results interpretability. We applied BiCoN to the TCGA breast cancer dataset, and we expected BiCoN to be able to recover known subtypes assigned with the gold standard PAM50 gene panel. For the analysis, we focused on patients with the most common molecular subtypes luminal A, luminal B, and basal. As a proof of concept, we first showed that BiCoN can separate patients into the 2 clinically well distinguishable subtypes, luminal and basal. As expected, the separation between patients with luminal and basal breast cancer subtypes is straightforward, and the clusters corresponds to the subtype labels. Next, we consider only patients that were originally classified as luminal to see if we can further stratify them into subtypes luminal A and luminal B, which are known to be difficult to separate on the level of gene expression. Here's our solution, and as you see, it does not completely agree with the PAM50 classes, but it actually agrees with something else. And the something else is the difference in cell type composition. We use the signature-based deconvolution method XCell, and in the right you can see that clusters reported by BiCoN show significant differences between cell type scores. And the derived subnetworks show robust correlation with the immune system response function, and that's reasonable to expect given that tumor samples are infiltrated with leukocytes. This demonstrates that BiCoN can find an interpretable difference between groups of patients even without being explicitly asked for it. The next question is if BiCoN is better in this task than other unsupervised methods. And here we can see that BiCoN performs as well as other methods like hierarchical clustering, but also finds subnetworks which explain subtype differences with a vastly number of genes than classical clustering methods. And these subnetworks offer a direct mechanistic explanation of difference between subtypes. Finally, if you would like to know more details about the algorithm and our results, please read our preprint or just ask me. Also, BiCoN is available as a web interface and a Python package. I'd like to thank my co-authors for their valuable contribution, and in particular Jan Baumbach, Tim Kapowski, and Markus Liest for their supervision. I also would like to acknowledge the Bavarian State Ministry of Science and Arts, as well as the Bavarian Research Institute for Digital Transformation for financial support. So, once again, in conclusion, BiCoN can cluster patients and extract subnetworks that can mechanistically explain the patient stratification. Moreover, simultaneous clustering of gene expression and networks makes BiCoN robust to noise and more robust to batch effect than typical clustering and biclustering methods. You can follow the QR code to get to our webpage. Thank you for your attention, and do you have any questions?